Okay, moving forward to the next technique. The next third technique is blue-white selection. This is also a very important technique and widely used in the genetic engineering, and you need to understand this technique very clearly because this is one of the favorite questions of the boards. They are every now and then they are asking about what is blue white selection, how will you identify whether this is transformed cells and non transformed cells. So this whole technique in this whole chapter of genetic engineering, these two things are very frequently asked: blue white selection and insertional inactivation. Okay, so let us understand what is blue white selection. Looking at this diagram, first of all, I want to tell you here that blue-white selection is also based on insertional inactivation method. What we are doing here also is inserting something for the inactivation of a particular gene. And which gene we are disrupting here is a lac Z gene. You need to understand and remember lac Z gene. This particular gene, lac Z, codes for beta galactosidase enzyme, and this enzyme, if produced, will act on certain substrate, and this beta galactosidase acts on X-gal. X-gal is a substrate on over which this acts, and they break down the X-gal. These enzymes break down the X-gal into a blue colored compound. Okay, clear? This is the basic principle on which this blue white selection is based. I'm repeating it again. Understand, there is a gene lac Z present. This lac Z gene is responsible for the production of beta galactosidase enzyme. Now, this beta galactosidase enzyme is acting on a substrate named as X-gal. This X-gal is broken down by these enzymes, beta galactosidase, to produce a blue colored compound. Okay, so this blue colored will be produced only when lac Z is active. That means lac Z gene is expressing itself. Clear? Okay, now coming back to the technique. This is the plasmid vector. Red color is the foreign DNA. What we have done, just understand. When the vector DNA, plasmid vector, has no insert present, I am starting from the third diagram here. It is not having, can you see anything red here? No. So it does not have any foreign DNA inserted. When it is allowed to grow, that means this light blue color is coding for lac Z. This light blue color of lac Z is very much active. So it will produce the colonies which are producing beta galactosidase, and when these colonies will be grown in a medium containing X-gal, they will break down X-gal and will produce blue colored colonies. Okay, so here see blue colored colony is being produced. That is what I was telling you, the basic concept. This happens. Normally lac Z is expressing itself and producing blue color colonies. Now the second case, what is being done here? The foreign DNA has been inserted somewhere in the plasmid vector, but this lac Z is intact. We have not disturbed the lac Z gene. Okay, so lac Z gene will be expressing itself in the second diagram also, and when it will be expressing itself, then ultimately it will produce beta galactosidase, which will act on a medium containing X-gal to produce blue colonies again. So blue colonies here also and blue colonies here also. These blue colonies are produced from both the types of cells. These are the non-transformed cells, that is which do not have the DNA insert, and these are the transformed cells which have the DNA insert. But how can we differentiate between them? We cannot differentiate because both are giving rise to blue colonies. We cannot differentiate them on the basis of color because whatever we see, we can differentiate easily. So here we cannot differentiate between them. So it is of no use. So what the scientists have done, they have incorporated this foreign DNA within the lac Z gene only. See here, the blue colored LAC-Z gene has been interrupted by the addition of foreign DNA. Now what will happen? This LAC-Z gene will not be expressing itself. Ultimately, the beta galactosidase will not be produced, which will finally affect the X-gal, and X-gal will not be broken down into a blue colored compound. Okay, so what will be the ultimate result? The ultimate result will be white colored colonies. Clear? So white colored colonies are easily visible. You can actually differentiate between blue colored colonies and white colored colonies, and by this whole technique we can understand, we can find out whether the transformed cells are there or not. See here, we cannot differentiate. In these two cases both are producing blue colored colonies, so we cannot differentiate although the transformation has taken place. But this transformation is of no use because we cannot select the transformants from the non-transformants. So what we have done here, we have incorporated the gene of interest in the LAC-Z gene only. So the LAC-Z gene is not expressing itself, and X-gal is not being broken down, and no blue colonies are formed. So what is the color of the colonies which are being formed? They are either, we call it as yellow, they are actually transparent or light dirty whitish kind of colonies. I'll show you the picture. See here. Here, this is culture in which you can see the white dots and the blue dots. These blue dots are mixed. Blue dots can be transformed cells also and can be non-transformed cells also. But can you differentiate? No, you cannot differentiate here. But when you look at the white colored cells, you can obviously differentiate between blue and white colors, and then can select only the transformed cells for further experimentation and taking out the product from the transformants. That was our main aim of actually doing all this genetic engineering thing. What was our main aim? Our main aim was introducing the gene of interest into the host cell so that every time the host cell replicates, divides, reproduces, it produces the many copies, it produces the product of our interest also. So how that aim will be fulfilled? It will be fulfilled by extracting the transformed cells and utilizing further. Okay, so see, this is a very good image of the culture medium, and you can see the blue white selection. This diagram has to be drawn. This is a very important technique. You have drawn the diagram. The white colored colonies are white, that means you have not colored it with the black color pen, but the blue colored colonies you have to fill it with the blue color pen so that it is visibly blue in color. Okay, so this is a very very important concept. You need to understand, learn these things. So I think this is all for today. I have told you about the agrobacterium mediated transfer and direct and insertional inactivation method and blue white selection. Just learn the notes, try to understand, watch the videos again and again for better understanding, and then we can move forward. We can discuss further. There's only few topics are left in this chapter. We shall be continuing and completing probably this chapter in the next class only. Okay, thank you so much. All the best.